The overall goal of this procedure is to perform transcranial magnetic stimulation to elicit motor-evoked potentials, first in the flexor carpi radialis muscle and then in the erector spinae muscles. This is accomplished by first preparing the skin overlying the respective muscles for electromyographic recordings and biomechanically positioning the subject for transcranial magnetic stimulation. The hotspot is identified as the portion of the stimulation location over the motor cortex that elicits the largest motor-evoked potentials. The next step is to determine the motor threshold. At this point, single-pulse transcranial magnetic stimulation is performed to quantify motor-evoked potential amplitude and silent period duration. Paired pulse transcranial magnetic stimulation is also performed to quantify intracortical facilitation and short and long interval intracortical inhibition. Ultimately, in vivo indices of corticospinal and corticocortical excitability in humans can be assessed via combined use of transcranial magnetic stimulation and electromyography.
Generally, individuals new to this method may struggle because it can be difficult to derive and understand the subtle technical aspects of TMS from a written description only, and simply attempting to learn through a trial and error approach when experimenting with and testing human subjects poses many difficulties and challenges. Demonstrating the procedures, in addition to myself, will be David Goss, Jr., a researcher working within the Ohio Musculoskeletal and Neurological Institute.